Okay, I totally just made most of this video and messed up pausing it, so I got to restart. So I'm gonna go through this pretty quick because I've already taught it already. Okay, so first thing I'm gonna go over is taste. After I go over taste, then I'm gonna go over smelling, vision. Then I'm gonna go over receptive fields, and then hearing. So all the senses. Okay, we're gonna talk about like the chemo path, so chemical. Transduction. So this is like sensory transduction. So when you turn a chemical stimulus into actually something the brain can perceive. Okay, so the first thing I'm going to talk about is going to be taste again. So last time I was doing this, I actually drew out a tongue right here. I don't know if we can see that. Yeah, we can. Okay, so I drew out a tongue. All right, and this is called the fungiform papillae. And the reason I mention this is because actually underneath here are, hundreds of these things called taste buds. Okay. And these are the things that actually will help in the process of us understanding what we're tasting. And so if I were to draw over here this little taste bud, we would know that there's, about 120 cells of these. Okay. There are also epithelial cells. Okay, the reason I bring up epithelial cells is because they don't have action potentials. Okay, so that's really important to know. These are graded potentials. The next thing that we want to know is that we also have basal cells. Okay, and what these basal cells do is they replace worn out or dead tissue of these epithelial cells. Okay, and they become the new gas station cells for tasting. So something that I also wanted to mention was the 5 different things that we taste. So the first thing is salty, right? This is going to be sodium. The second thing that we're going to know that we taste is going to be sweet, sour. Okay, these are acids. Acids, and so these are going to be hydrogen ions. Okay. Acids. Okay, the third thing is going to be sweet. These are going to be carbs. Okay, sweet carbs. Okay, fourth thing is going to be bitter. Things that we taste that are bitter. Things that are bitter are going to be calcium. Okay, and the fifth thing, and something from organic chemistry as well, but we'll skip that. And the fifth thing is going to be so salty, sour, sweet, bitter, and then we have umami. Umami. Okay, with umami, this is glutamate. Okay, so remember how we talked about how these are taste buds and epithelial cells? They don't actually carry out an action potential. So what do they do exactly? Well, we'll go over that. So I'm going to draw out a couple because there's 3 that we really want to know with these 5 different tastes that we can taste, okay? I don't know if we can see that. Maybe that looks okay. How's that work? Okay, perfect. So I'm going to draw out 3 different cell bodies here, okay? Number 1, number 2, number 3. Okay, the first one is going to be taste receptors. Okay, taste receptors for salty things. So that's sodium. Okay, the second thing is going to be taste receptors. Okay, for sour things. That's going to be those hydrogen ions. Okay, the third thing is going to be taste receptors for 3 different things. We're going to have bitter, which is going to be our calcium. We're also going to have sweet, which is going to be our carbs, glucose. And then we're also going to have umami, which umami is going to then be glutamate, right? All right, so these pathways and the way that they conduct sensory transduction, where they go from a chemical stimulus to then telling our brain, oh, you're tasting something salty, or you're tasting something fatty, you're tasting something delicious, umami. The way that it does that is through these processes. Right here on the membrane, for salty ones, you would have a sodium ion channel. When the sodium is tasted in the taste bud, we end up causing this type of depolarization within a graded potential in the cell. This depolarization actually causes something to happen on the membrane. We have calcium gated ion channels. If we have a strong enough depolarization, not to action potential, but strong enough to actually sphere of influence to this channel, we actually have a release of calcium within the membrane. We have vesicles in here. This is important because there's T snare and B snare proteins, so then this actually ends up turning into something like this, where we have a release of a neurotransmitter to our then neuron that will carry this out to the brain with a receptor on it. So what we end up seeing here is something like this. Calcium comes in, helps bind with the snare proteins, thus facilitating exocytosis of neurotransmitter, and we taste something salty. In hydrogen ions with something that we would call sour, right? If we had something sour going on in our mouth, we would pick it up through this process. What would happen is this comes in, hydrogen ions come in, and we get some sort of depolarization as well. Now these ones, we have a channel over here called the potassium leak channel. Okay? With the potassium leak channel, potassium wants to come out, but what does hydrogen do? It actually wants to come in right over here, and so sometimes it can actually get in the way, and it actually can block this. If that's blocked, then we know potassium raises up in concentration within the cell. What does that do? It causes a little bit of depolarization. We have a sphere of influence here within this calcium gated ion channel. Calcium comes in, same thing. We have these vesicles right here. If we have calcium inside the cell, then these can fuse T snare and B snare proteins. We actually have exocytosis, release, fusion of membrane, and vesicle exocytosis of neurotransmitter. Okay? And then we taste something sour. How it works with umami, bitter, and sweet is something like this. Here we have a receptor on here. Okay? We have a receptor right here. If we had something like glucose, something sweet come in, we just ate a cake, glucose comes in, binds right here on this receptor. There's a cascade of events that occur here, and then this actually activates something called a phospholipase. Let me see if I read that right. Yeah, that's right. Okay, phospholipase. So phospholipase gets activated. When phospholipase gets activated, it actually breaks something down into two products. One, and the second one, I actually know this one's IP3. So we don't really think about the first one that much because IP3 is the most important thing in this case. In the cell, we have the SCR, smooth endoplasmic reticulum. What do we know about that? Calcium concentration is very high. Calcium concentration is high in here. IP3 can come over, bind here, and this is some sort of ion-gated, ligand-gated ion channel, so calcium can then come out, and then we have high concentration of calcium coming out of the cell, and we get the calcium that we need. We also have calcium leak channels on this side as well, and these calcium leak channels can allow calcium to come in the cell as well. So we have IP3 in this case for bitter, sweet, and umami, and then we also have these calcium leak channels. These again cause these vesicles to fuse with the membrane. When they do fuse with the membrane, we actually have this release of neurotransmitter. We have the neuron here. Neuron gets the signal, sends it to our brain, says, that's pretty tasty. Okay, so the big thing to remember is salty, sodium's coming in. Sodium actually changes, allows calcium to come in. Hydrogen ions are coming in. They're actually blocking the potassium channels from leaking, and that raises the depolarization to allow calcium to come in. Then sweet, bitter, and umami, they're actually doing a this GPCR, or GCPR, so G coupled protein receptor. Forgot to mention that that's what that is. This causes a cascade of events, thus activating phospholipase. Phospholipase then will cut something in half into IP3. Given us the product of IP3, we can then activate a ligand-gated ion channel on the sarcoplasmic, or on the smooth endoplasmic reticulum. This will actually allow calcium to come out of the SCR into the cytoplasm and thus lead to a extracytosis of vesicles, because then those T snare, B snare proteins can bind via the calcium, exocytosis through fusion, and the release of neurotransmitters so we can know what we're tasting. Okay, that's gonna be taste. Okay, so smell. I'm gonna go through this pretty quick. I'm gonna try to draw this the best I can. For smelling, what we know is we kind of have our nose, and then we have this palate here, right? So this is our hard palate. This is going to be our nose here, right? This is our smelling. This is going to be our nasal cavity here, right? Nasal cavity. Okay. This is going to be a particle of methane gas that we end up smelling. So this is CH4. This comes in. We have some sort of vesicles here, and that releases up to here. And then up here, we have something called the cuneiform plate. Okay, this is called the cuneiform plate. A little bit of anatomy. Okay, this has these spaces here, separation for these neurons to come up and through. Right here, we have a like a sinus membrane. What do you call it? I would call it what did I call it? Oh, mucus layer. Okay, yeah. So this is going to be your mucus layer, right? Easy enough. Mucus layer. That's your mucus layer, okay? If that's your mucus layer, what ends up happening is you have these neuron cells that kind of go like this. Okay. It's a little bit less accurate than this, but you know, you get the point. These gases are going to come up, bind to these receptors on these nasal cells here. Okay. You also have epithelial cells in here with nuclei there. These are called epithelial cells. Okay. So one thing that's important to note here is that all of these would come into an olfactory neuron here. Neuron, okay? And then up here, we would actually have a structure here called the olfactory bulb, okay? So, what we want to, let me make sure I didn't miss anything here, okay? Okay, we're good. Okay, so if we were to take something like this, okay, we want to take one of those cells that are in this mucus layer and they're picking up these chemicals, they're picking up these methane molecules here. And we want to zoom in on this a little bit and kind of see how this works. Well, if this was the cell, okay, here, the body, and we had a receptor up here, okay? Same as with the taste, this is going to be a G-coupled protein receptor. So we know that there's this cascade of events that need to follow based off of its function. So G-coupled protein receptor, what ends up happening is this is going to go through a couple things. So let's say up here we had methane bind to this active site here. We have a gas that binds. We get a cascade of events. This actually activates something over here, okay, called adenylate cyclase. Adenylate cyclase. Adenylate cyclase. When adenylate cyclase is activated, what this does is it comes down and it can bind on a special channel that brings sodium and calcium in, I'm pretty sure. Okay. So with olfactory, this creates adenylate cyclase. When adenylate cyclase is created, this is actually going to be known as CAMP, okay, cyclic AMP. When cyclic AMP is thus converted, there's a binding site for cyclic AMP on this protein channel. What ends up happening is you have increase of sodium coming in through this channel, that's activated, and you also get your potassium that comes in through this channel. But you also have chlorine ions that are coming out of this channel, okay? So chlorine is coming out, sodium and potassium are coming in. What we end up seeing here is we get this depolarization within the cell. And it becomes more positive, right? And if it becomes more positive, then we're able to open up these calcium channels, okay, which then allow for us to bring calcium in, and then we can have this fusion of the membrane to release neurotransmitter. To those olfactory neurons, okay, which then carry signal. Okay, so just to recap everything, what's going to happen okay, I actually got this wrong right here, and this is not needed on this one. Okay, so right here we would have actually calcium coming in through this channel, okay? Okay, so let's review this real quick. So what ends up happening, you have a methane gas comes in through the nasal cavity. When it comes through the nasal cavity, it's gonna go to the mucus layer. If it's in the mucus layer, it ends up going through this process to then get up through the olfactory neuron to the olfactory bulb in between the cuneiform plate. You also have epithelial cells here, but this is taking place within these olfactory cells that are going to be actually uptaking things in our environment to then make sense of what we're smelling. How this part works here. Methane or a molecule will bind to a receptor on a G-coupled protein receptor. This causes a cascade of events. When the cascade of events proceed forward, we end up seeing that because this methane molecule bound to the G-coupled protein receptor, we then have this, it would almost be called transducin as well. But what ended up happening is you have adenylate cyclase, and adenylate cyclase is activated. Then adenylate cyclase is an enzyme, and then that will actually produce cyclic AMP, which is CAMP. CAMP then goes down and will bind. Okay, it will bind to a special pump and then turn it on, and this pump then takes sodium into the channel and calcium and removes chlorine. If you take calcium into the channel and sodium in and you remove chlorine ions, we end up depolarizing the cell, and we actually allow for the T snare and B snare proteins to grab onto each other, pull that vesicle to the membrane, and allow the release of neurotransmitter into the synaptic gap to then cause a action potential to then reach the olfactory neuron here, going up through the olfactory neuron to the olfactory bulb. Okay, so again, one more time, I'm going to walk through this part because it's really important with CAMP. What's going to happen is you're going to have that sensory within the environment, a methane gas, for example, bind to the G-coupled protein receptor. When the G-coupled protein receptor receives that stimulus, it's then going to cause a cascade of events. Now we know with taste, with bitter, sweet, and umami, they're going to use phospholipase. And then phospholipase then creates IP3, which then turns on the smooth endoplasmic reticulum. Don't get those confused, because in this case, we're going to be using CAMP. So cyclic or adenylate cyclase will then turn into cyclic AMP because it's an enzyme. So we have CAMP. CAMP binds that special receptor. That receptor brings in sodium, it brings in calcium, and it removes chloride. When it does that, we depolarize inside the cell and we send off the vesicles because we have the calcium that we need to allow for the fusion and exocytosis of the neurotransmitters. Once those neurotransmitters are released, they end up going to the olfactory neurons, which then feed up into the olfactory bulb, which will then go to a part of our brain where we'll make sense of what we're smelling and use that information to make decisions. Okay, that's going to be smell. Okay, so vision. Let's go over a little bit of anatomy of the eye, okay? So how I want to do this is I'm going to go over the anatomy of the eye and then some stuff with the lens, and then we'll get into how phototransduction actually works, okay? So this is how we're going to do this. With vision, we have the eye, right? Here's the eye. We have these protein muscles here. Okay. Then we have our suspensory ligaments. Okay, perfect. Okay, then we have something right here. This part here. We have this leading back to here. Okay. Kind of a weird looking eye, but it's the best we got. Okay, this right here is going to be our cornea. This part sits right on top of the eye. This helps with a lot of refracting as well. This is going to be the iris. This is going to contain those special muscles that will help dilate and relax the eye for the pupil. This spot right here that you see that opens up dotted, this is going to be our pupil. If that's our pupil, we know that this is aqueous humor in here, and then vitreous humor on the inside of the eye. Okay, this right here is going to be called the ciliary muscle. Very important when we talk about the lens. And then this is going to be called the suspensory ligaments. Okay, if those are that, then this is going to be our lens. Okay, lens. If that's the lens, this is the post area back here, and then this is going to be the anterior up here. Okay, this is going to be our optic nerve. Okay, that's our optic nerve, and then this is going to be called the retina. Okay, this is where the phototransduction actually occurs. Okay, very, very important areas. So, if phototransduction occurs right here, what we want to know is what are the layers of the cells that are here? So we have photoreceptors. Okay, so we know that. We got photoreceptors. We have the bipolar cells. Okay, we got bipolar cells, and then the third thing we have in here are going to be our ganglion cells. Okay, so we got our photoreceptors, bipolar cells, and the ganglion cells. Up here with the lens. Okay, let's take the lens up over here. We got a couple things that we need to talk about in terms of the lens. So we have two different types of structures with the lens. We have convex and we have concave. Okay, can we see both those? Convex and concave. Okay, so what ends up happening? You have a convex lens. This means that the light, okay, light is pointing inward. So if we shine light at it, at a convex sphere, it would direct the light inside. And I believe this is when your focal point is too far in front. We can push it back by directing it backwards because we want the light to hit the retina, okay? The next thing about light is, so the light is going to bend inwards. That's a better word, bend. The light will bend inward, okay? The other thing we want to know about convex is the ciliary muscles ciliary muscles are going to actually contract. This actually causes bowing within the suspensory ligaments. Okay, if we have bowing within the suspensory ligaments, then we actually have this squeezing of the lens, and so this all has to do with the lens. Okay, and so what we end up seeing is we can see things better up close. Okay, let me make sure I got everything there. I only forgot one thing, and that is we want a thick lens here, okay? In contrast to that, we have convex and we have concave. So the light bends outward. Okay, bends outward. Light's gonna bend outward. When the light bends outward, we actually have relaxation of those ciliary muscles. Okay, if those are relaxed, then we actually stretch. Okay, okay, so those suspensory ligaments are going to be loose. Okay, suspensory ligaments are going to be stretched. Okay, when we're relaxed, they're going to be stretched. So it's going to pull on that lens. So these are going to be stretched. Suspensory ligaments. Okay. If the suspensory ligaments are then going to be stretched, then we know that the lens is going to be thin and long. Okay? Thin and long, and then this will help us see far away. All right. See far away. So if we can see far away, then we're going to concave. So concave, we're gonna loosen those ciliary muscles, which is going to stretch those suspensory ligaments, which is going to pull that lens into more of a football form, squeeze it up, elongate it so we can actually see further. It's going to refract light outward. Okay, and that's about it for that one. Okay, and then it's just going to help us see far away. Okay, so we got those two things down. Now we want to talk about how sensory transduction is actually going to occur. We call this phototransduction. Phototransduction. Okay, phototransduction. Something that we want to know is this. There's those 3 cells that are involved. I'm gonna have to draw it off to the side here. Okay. Okay. First thing I'm gonna do is draw it with this for light on. For light with photoreceptors. Okay? So this would be cones, rods, so all gonna kind of work the same. Okay, so the first thing I'm going to do is draw it with light. Then I'll draw on this with another color for another thing. Okay, let me make sure I got this. Yeah, we're good. Okay, one other thing I want to mention is up here, we actually have on the layer is this epithelial cell. Okay, they actually help with absorbing unwanted light. Okay, absorbing unwanted light. So these are going to absorb that unwanted light, right? What ends up happening is this. We have a cascade of events that are going to occur. Light comes in to the picture. On the membrane, we have a protein right here called so we actually have rods and cones over here. Rods and cones. Okay. Rods are black and white. Black, night vision, dim light. Cones are going to be light, color, it's 1:1. This is many to one. Color, we're going to see green, blue, red, okay, and a couple other things. Let me look really quick here. One other thing, I don't know if this is necessary, this is scotopic. Scotopic vision. This one is photopic. Pretty sure that's right, photopic, yeah. Photopic vision, seeing color and those kinds of things, okay? So then you can see color and whatnot. So this is going to be light emitted, right? So in this case, we're talking about a cone, okay? Light's going to come in. On the membrane, we have a molecule. Trying to remember the name of this one. Is it rhodopsin? I'm pretty sure it's rhodopsin. Oh, that's easy. Yeah, I know this one. Okay, so this is a G-coupled protein receptor, okay? G-coupled protein receptor. Okay, G-coupled protein receptor on the membrane. This G-coupled protein receptor has a molecule within it that's called cis-11 rhodopsin, retinol. This one uses rhodopsin. This one uses iodopsin. Okay. Iodopsin. Okay. So you have GCPR, G-coupled protein receptor on this membrane. You have cis-11 retinol on here. Cis-11 retinol, when it gets hit with light, turns into all trans 11 retinol. Right? Let me see. All trans retinol. Okay? Turns into all trans retinol. When this turns into all trans retinol, it causes the alpha subunit to release, and then this whole process is called transducin, and then this will all activate PDE, phosphodiesterase. Right? So that's going to produce phosphodiesterase. Sorry, I just want to make sure I'm spelling this right. PDE, phosphodiesterase, this is an enzyme. This enzyme is within the cell, okay? And I'm going to draw it as this little Pac-Man here, like this, okay? So you have phosphodiesterase just like that, okay? Phosphodiesterase actually does something to CGTP. It's going to, well, cyclic GMP. CGMP, this eats it up. On the membrane, we have a protein here, channel, what this does is this protein channel is going to allow, what's it called? So on this protein channel, we have sodium coming in on it. And CAM actually activates that leaving of that material. If we eat up cGMP activates the input of this material. If we block cGMP, what we end up doing is we end up depolarizing the cell. We have IPSPs, inhibitory postsynaptic potential. And so this actually inhibits. Why is that good? Well, here at the synaptic gap, that means we will release low amounts of glutamate. This is important and good because on this membrane, we have a protein called mGluR6. mGluR6, when it gets activated by glutamate on its receptor, it then has a cascade of events and messes with TRPM1. This is a calcium channel. If we mess with TRPM1, what mGluR6 does is if it gets a lot, it can block that. In our case, we don't have a lot of glutamate, so we don't have a lot of mGluR6 being activated to then block or mess with TRPM1. So then calcium levels are low, or would be high, because it's not being inhibited. So we have a lot of positive charge coming in. Thus, we also have a lot of calcium to help bind with vesicles for the actual fusion and release of neurotransmitter glutamate. So then down here we can say we have high levels of glutamate being released. If we have high levels of glutamate being released on the ganglion cell, and I actually forgot to label these, this one's going to be the photoreceptors, this one's going to be the bipolar cells, and this one is going to be our ganglion cells, where our action potentials can occur. So what we end up having is, this is called AMPA. This is an ion-gated channel. So when glutamate binds to here, this is ligand gated, so then we allow ions to flow in. And if we have a lot of glutamate, then we can get this high positive charge within the cell. So this actually is going to be a depolarization. And that's what we get here is depolarization. And if we have depolarization in both those spots, we actually have action potential. And this will go down the optic nerve. Okay? That goes down that optic nerve. Here's the cool part. In some cases, and in this case, okay, in this case, this is in the case of light. In the case of dark, notice I'm using the black marker to illustrate darkness, okay? We inhibit the light. No light, we don't change cis-11 retinol to 11 trans retinol in rhodopsin. Transducin doesn't occur. We don't activate phosphodiesterase, so we don't eat up cGMP. If cGMP is not eaten up, then that binds to the ligand-gated channel and allows for this positive charge to come in. Okay? Yeah, I knew it. This is sodium and calcium. So this is sodium and calcium. Okay, they both come in. If we increase calcium and the sodium channels coming in because cyclic GMP is now, cGMP is now available in the cell, we then allow positive charge to come in to the cell. I should be writing this in black for you. Okay, we then allow this positive charge to come into the cell. If we have positive charge within the cell, we know that that is going to actually depolarize the cell. If we depolarize the cell, then what ends up happening is depolarization occurs. So then if depolarization is happening here, then we have a large release of glutamate. Okay, if we have a large release of glutamate, then we actually have a lot binding to mGluR6. If a lot are binding to mGluR6, then that means we're inhibiting this TRPM1, this calcium-gated ion channel. If calcium-gated ion channel is being inhibited, then we don't have a lot of release of our neurotransmitter. We actually have a inhibitory step here, and this would be called inhibitory or hyperpolarization. And then we have small release of neurotransmitter glutamate, and that feeds into AMPA very little, and therefore we have no action potential in the ganglion cell, which goes to the optic nerve. So in darkness, that's why we don't send any signal. Okay, so that's light and dark. Let me make sure I got everything on my notes here. Cool. All right, well, I think I got everything. So let me just walk through it really quick one last time. We have convex, concave. Concave or convex, it's going to point light in. It's going to be to see things up close. With concave or convex, we also have a thick lens to see those things up close. If we have a thick lens and we're going to have contraction on the ciliary muscles, and what that does, that contraction down actually puts a bowing within those suspensory ligaments, thus allowing for the squeezing of the lens. That's why it gets thicker, and that's why it points things, bends the light inward. Concave is going to be actually refracting the light outward. If we refract the light outward, then what concave is going to do is concave is going to, let's see. So concave is going to be a thin, long, elongated lens, kind of like a football. And then the suspensory ligaments are going to be stretched because we have relaxation of the ciliary muscles. And this is to be able to see far away. Let me make sure I got that. Cool. Then when we get into the anatomy of the eye, we actually focus back here on the retina. We focus on photoreceptors, bipolar cells, and ganglion cells. As I was walking you through, we have light that comes in on a photoreceptor for like a cone, okay? That, well, see color and light, but so let's just say for one that sees light, okay? If light's on. If light's on, light's gonna come in. It's gonna hit the GCPR, G-coupled protein receptor. When it hits that, it actually transitions cis-11 retinol to all trans retinol. When all trans retinol is activated, it actually releases an alpha subunit. That alpha subunit will go on and activate the, that will go on to activate the phosphodiesterase enzyme. If phosphodiesterase is activated, then cyclic GMP, CGMP, is going to be eaten up in the cell. CGMP actually binds to receptor on a channel that allows sodium and calcium to come into the cell. If that CGMP is missing, eaten up by phosphodiesterase, then we end up having that channel closed off. So then it's inhibitory. So we have hyperpolarization. So we have at the end a little bit of release of glutamate. That little bit of glutamate being released actually is a good thing in this case, because on the bipolar cell we have a receptor, and that is called mGluR6. mGluR6, its job is when it binds to glutamate, it's going to actually inactivate this channel on the bipolar cell that allows calcium to come in, and that's not a good thing. And so it's called TRPM1. And so in this case, when we have low amounts of glutamate, we're not going to inhibit that. We're actually going to leave that open and let the calcium come in. So when calcium comes in, it's actually really, really good, and allows for the fusion of the vesicles with T snare and B snare proteins. And then we actually have the release of neurotransmitter, more glutamate, in between that synapse between the bipolar cell and the ganglion cell. When the ganglion cell then gets that glutamate, it actually is going to cause action potential to occur, depolarization, allowing those sodium channels to open up and depolarize the ganglion cell, thus sending an action potential and then through the optic nerve. Okay, so one other thing to remember is that we don't have any photoreceptors where the optic disc is at in the eye. Now, the last thing I'll cover really quick is in darkness. In darkness, we inhibit the light. If we inhibit the light, we actually open up the, we keep cis-11 retinol. We don't change to trans-11 retinol. If we don't change to all trans retinol, then we don't activate through this transducin. We're not going to have alpha subunit go and activate phosphodiesterase. If phosphodiesterase isn't activated, then we don't have, we have high levels of cyclic GMP, CGMP within the membrane, or within the cell. So if we have high CMP, that's going to bind to that receptor over here. There's a receptor over here, and that will allow in a lot of sodium and calcium to come in the cell. If those come in, we depolarize the cell. We send out a ton of glutamate. If we send out a ton of glutamate in between that synaptic gap between the the photoreceptor and the bipolar cell, then we're going to end up sending off, activating a lot of mGluR6, which will block a lot of TRPM1, and we're going to shut down calcium in the bipolar cell and cause depolarization. So we have minimal release of neurotransmitter glutamate in between the synaptic gap of the bipolar cell and the ganglion cell. And what that ends up doing is that sends an inhibitory response, IPSPs, and we get no action potential, and so we don't see anything. So that's going to be how that process works, okay? This one's just going to be, so many channels to keep track of. Okay, so one of the last things I want to hit on is going to be receptor fields, okay? Receptor fields. Okay, receptor fields. What this is trying to depict is, we got a couple rules that I want to go over here. A couple rules, all right? For the rules, what I want to cover is that Um, light means low glutamate. Okay, light means low glutamate. On, let's see, how do I phrase this? If it's on, hang on, how did I say this before? I think it's light and on equals action potential and darken. Yeah, I don't know. These are the rules I really like to follow. If you have on center, then your bipolar cell is going to be, the receptor on it is going to be mGlu-R6. Okay? mGlu-R6. If the is off center, then the receptor on here on the bipolar cell is going to be off center, on center, off center, mGlu-R6 AMPA. Okay? So I'm going to draw a couple cells here. Okay, our first one will be right here. This will be our second one. And our third one. Then we'll have a bipolar cell here. And then we'll have our ganglion cell coming this way here. Okay? Does that make sense? Photoreceptors, bipolar cell, ganglion cells. Okay? Cool. In this case, we're going to call this on center. Okay? And off surround. Okay, this is going to be 60%, 20%, 20%, and we'll get to those, maybe at the end, but that's kind of more for different practice problems and things that we can be given. Okay, so on center, off center. This has to do with light, okay? If it's on or off, has to do with light, okay? So what this is saying is if light is hitting this, it's gonna be off. If light's hitting this, it's gonna be on, okay? Let me walk through a couple scenarios, okay? We just went through this. If we had light come in, okay, we hit G-coupled protein receptor. When we hit G-coupled protein receptor, we'd go from 11 cis 11, right? 11 cis retinal to all trans retinal. When we go to all trans retinal, okay, when we go to all trans retinal, what we end up doing is we allow that alpha subunit, okay, to then activate phosphodiesterase enzyme. If phosphodiesterase is then activated, then what ends up happening is we break down and eat Mr. Pac-Man here, cyclic AMP or CGMP. Okay? More. This is on the receptor here. We have sodium and we have calcium coming in. But this inhibits it if we don't have this to do that. So then we actually get a depolarization in the cell, IPSPs. Hyperpolarization, I mean. this comes down. We have a negative charge coming down the cell, right? When we get to the end, that's going to release low amounts of glutamate. If on this membrane we have mGluR6, in this case it's on center, and over here is going to be our TRPM1. If mGluR6 isn't activated that much, TRPM1 isn't going to be inhibited, and this is going to be our calcium channel, right? Come in to the cell, and if calcium can come into the cell, then we're going to have more release of neurotransmitters. Okay, we just went over that, right, in the last part. This leads to what's called an action potential within this ganglion cell, which will then fire to the brain. So that's why we call it on center. When light's hitting the center, it's on, okay? It's firing an action potential. Now let's try this one over here. Let's try this one in blue. Off surround. If light hits surround, it's going to be off. The reason is, light hits this, we have a low release of glutamate, right? In between these two cells, something I failed to mention is we actually have these things called horizontal cells, okay? Horizontal cells. Okay? So if we have horizontal cells right here, what we see is low glucose levels. On these horizontal cells, we have a membrane protein called AMPA, right? Well, if glucose is low, AMPA is going to be low activity. We then have a release of GABA as a neurotransmitter. So if those are low, then GABA is low. GABA actually allows chloride ions to come into the cell and allows for potassium ions to leave the cell through a leak channel. This actually causes IPSP within here. So what this ends up doing, the reason it's off is because it doesn't end up doing that. It actually makes this more positive, which actually sends tons of glutamate. If we send tons of glutamate, what does that do to mGluR6? Well, mGluR6 is going to be fired up. mGluR6 is going to actually activate a lot of blockage inhibition on TRPM1, which then allows calcium to not come in, which then allows no action potential. So that's why we can say that it's off, okay? Now over here on this side, I'm going to show what it's like in the dark. If it is dark, okay, let's say it's dark. What do we remember about dark? Well, dark inhibits the light, right? If dark inhibits the light, we stay as cis-11 retinol. We don't switch to all trans retinol. We stay at cis-11 retinol. Cis-11 retinol. We don't activate phosphodiesterase. We don't chew up cGMP. cGMP can actually bind to those channels where sodium and calcium can come into the cell, because we have cGMP that can bind here. And then we actually have this depolarization, EPSPs. If we have depolarization occur, this gets more positive as we come down. Then we have a lot of release of glutamate, right? If we have a lot of glutamate release, what does that do? actually it's up here, right? Then AMPA. AMPA is super high right here, right? So if AMPA is super high, then we have a lot of GABA released. If we have high amounts of GABA released, we have high amounts of Cl coming into the cell. If we have high amounts of Cl coming into the cell, we have high amounts of chlorine or potassium leaving the cell. If we have high amounts of that, then we end up having IPSPs. If IPSPs are here, we end up having an inhibitory, and we have the same as this, and so we fire an action potential. So then we can add another rule. Off plus dark equals action potential. On plus light equals action potential. I believe that's one of the rules I had on here. Okay, last thing I want to show is this. You can have another scenario, okay? Can we see those? Yeah, okay. We can have another scenario where it's off center, okay? All this means is this receptor is gonna change to an AMPA receptor, okay? That's all that's saying, but it changes everything, right? So off, surround, on, surround, become on, surround, okay? Off center, on surrounds, right? Okay, so that means that if light's hitting it, it's gonna be on. If light's hitting the center, it's gonna be off. But if darkness hitting the center, center will be on. So let me walk through this one really fast here. This is what's gonna happen. Let's show an off center and on surround, okay? Off center, we have darkness hit, right? Darkness hits, we inhibit light. If we inhibit light, then we know that through this whole cascade, we actually have positive charges being released. Then we have a high amount of glucose being released in the synaptic cleft, and that actually activates AMPA to cause positive charge. Then we have high amounts of glucose being released here, which then that activates AMPA here, and then we have positive charge depolarization leading to action potential within the ganglion cell. Over here, on surround. If light hits this, what do we know about light hitting it? We're gonna have low levels of glucose, glutamate, released, right? If we have low levels of glutamate released, then what does that do? Well, low levels of glutamate means we have low AMPA. I'm gonna use a pen. Means we have low AMPA, right? If we have low AMPA coming in, then we have low GABA here. So then we have low Cl coming in, and we have low potassium coming out. If those things take place, this is going to be positively charged. If that's positively charged, then we're going to have high amounts of glutamate. If we have high amounts of glutamate, AMP is activated, positive charges. If those are positive, we release a lot of vesicles. High glutamate. Here, we have positive charge, and we have action potentials that occur. Okay? So that's this theory on receptor fields and how they work. Perfect. Okay, last topic, easiest one of them all. We're gonna try to fly. Gotta fly through this. Okay. So this is what's gonna happen. Whoops. Okay, so auditory hearing, right? Okay, so we have this structure here called the ear, right? Okay, we've got a sound wave that comes in. This is going to be the eardrum, ear canal. This will be something important here that's called the tympanic membrane. Okay, tympanic membrane. Connected to the tympanic membrane are going to be a series of these little bones here. Connected on their edges, right? And then this is going to tap on something here, the oval window. So these are called the ossicle bones. Okay, this is going to be malleus, incus, stapes. Okay, this is going to be the oval window. And this leads into the cochlea. Okay, which the cochlea actually is going to be represented more like this, kind of like a snail. Okay? So, with auditory hearing, we have these chambers within here. It's called the scala vestibuli. It's called the scala tympani. This is called the scala media, but it's also called the endolymph. And that's the type of fluid that's in here. And this has perilymph. This is perilymph. Okay? Those all have perilymph. Sorry, bad video there. Okay, those all have perilymph. That has that. This is called also the cochlear duct. Okay, and this is also called the basal membrane. Basilar membrane. Okay. All right. So something that we want to know about this basilar membrane. All right, I'll explain how sound works going through this in just a second, okay? But what I want to do is draw out this structure right here. If we took a cross section of this, flipped it out like this, we would see that there's actually some compartments to this. called the Sterling. This is called the striae vascularis. Okay, striae vascularis. In the striae vascularis, you have these two parts here. The reason this is so important is because we're showing how this gets set up. Why the endolymph, right, is so important. Well, what we know is endolymph is going to have a high concentration of potassium, and these have high concentration of sodium, okay? And why? How does that happen? Well, we're going to walk through that. What ends up happening in these cases is we have the striae vascularis. Well, this has a sodium potassium pump on it, Na plus K plus ATPase, right? But it also has another pump on it, NK2Cl. So this brings a sodium in, and this is primary active transport. This is secondary active transport, and this brings sodium in, it brings calcium in, or potassium in, and it brings 2 chloride ions in. What we see here is that we actually have a leak for potassium ions. Here we have a leak for chloride ions. Okay. Then we move over to this next one. This one also has ATPase with a sodium potassium pump, primary active transport. This one being secondary active transport. We still have the NK2Cl pump, and that's going to bring in 1 NaC, Na plus, and it's going to bring in a C plus, K plus, potassium plus, and a calcium chloride, chloride minus 2. This is going to leave through leak channel. These leave through leak channel. Okay, let me make sure I got that right. Cool. Yeah, and that's why we have high concentration of potassium in this endolymph. Striae vascularis. So one thing I want to show is I want to zoom in on this spot right here. Okay. When we zoom in on this spot right here, what we find is we have these finger-like projections, right? And kind of like your hand or toes, they get taller as you go. On these, they have these membranous proteins on the membrane, and these are protein channels, and then they're connected by these link proteins. Up here we have this membrane, and these actually stick out into the membrane here. Okay, these hair-like projections, right? And in this membrane, this is called the tectonic membrane. Tectonic membrane. So what we end up happening, what we end up seeing with this if a vibration were to occur here, and I'll explain how this all fits together, but if we end up getting tapping vibration that occurs here, what we end up seeing is these hairs, these finger-like projections, they push upward, right? They push up. And as they push up on these hair cells end up getting bent, and they push this way. And they push this way. And they're connected. And so as they bend, they push. Now these are mechanically gated ion channels. And so these are mechanically gated ion channels, and what ends up happening is they open up as they move. And when they open up, we actually have potassium that's ready to flood in. Now this potassium floods in. And there's also, right here, we would have a calcium ion channel that allows for this to flow in, which then allows for the vesicle to then be able to fuse with the membrane and release neurotransmitter glutamate into the synaptic cleft to then bind with that neuron cell there. Okay, so these are snare, T snare, B snare proteins. They bind because of Ca++ coming in, actually facilitate that diffusion and fusion and then exocytosis in the synaptic gap. So then this calcium comes in, we have calcium right there. Okay, and then with high concentration of potassium, we actually allow it to leave down here through these potassium leak channels. And so this is the endolymph, this is the peri, and then we allow it to flow out. Okay? Okay, so let me put all this together. You hear a sound wave comes through, right? Sound wave comes through, goes through, hits the tympanic membrane. This hits the ossicle bones. We hit the malleus, incus, then stapes. Stapes taps on the oval window. This causes a fluid projection movement within the cochlea. If this is the cochlea, we have the scala vestibuli. Scala vestibuli is full of perilymph. Perilymph vibrates, we can go up and down, but it depends on where the frequency heads. This side is going to be low frequency. I'm pretty sure it's a high frequency. High frequency. So that's something important to note as well. This is going to be high frequency and thick and narrow. This side is going to be low frequency, thin and wide. Okay. Okay, so let me ask that again. So then down here, this is going to be low frequency, but it's going to be thick and narrow, and then up top it's going to be high frequency, thin and wide. And so this is important because then we know which signals, which cells, hair cells, respond to which different frequencies and signals that we're getting within the cell. So what ends up happening is perilymph vibrates, we end up pushing up on the basilar membrane. When the basilar membrane pushes up, we push those hair cells into the tectonic membrane. It's just like a gel-filled fluid, like Jello. Tectonic membrane pushes up, gets those and bends the hair cells. The bending pull on the linked proteins, which then open up the mechanically gated ion channels, allowing potassium to flow into the cell. When potassium flows into the cell, we depolarize the cell. Then we can allow calcium ions to come in. We have ATPase on the membrane as well, but the main thing is we have that potassium then leaving out from the endolymph to the perilymph. And then the process of how the perilymph or endolymph becomes very high in concentration of potassium is through the striae vascularis. Through the striae vascularis. And because of the striae vascularis, we then can have that high potassium levels in the perilymph. And so then we have potassium leave, but then calcium binds to the vesicles and can release those vesicles, bind to the snare proteins to allow the exocytosis of the neurotransmitters. So that's kind of how hearing works. And thank you, and that's all for the video.